So this one is a very important chapter in like the biochemistry and physiology. That's the molecular biology. So basically all the things we should know: what are nucleoside and nucleotides, DNA and RNA, then the enzymes which are like important in transcription and translation, mutations, and the other like the reactions and the clinical uses of the various techniques to diagnose the proteins, DNA, and RNA. So let's start first of all with this one. So remember, first of all, a nucleotide and nucleoside, the difference between nucleotide and nucleoside. So in nucleoside, we have only sugar and base. So there is no phosphate group. Whereas in nucleotide, we have a phosphate group. So in the base, we have two things, whether it's a nucleoside or nucleotide. It's a purine and pyrimidine. And in pentose sugar, it's a ribose sugar or deoxyribose. Which make difference in the nucleotide is the phosphate group, where there is additional phosphate group in nucleotide and it is absent in nucleoside. The next very important thing we should know is the purines and pyrimidines, the classification of the purines and pyrimidine. To remember this, we should remember pure silver. So for purine, pure adenine and guanine, and for pyrimidine, so it's cut the pie. So for pyrimidine, it's cytosine, uracil, thiamine. Very important thing, and we should remember by heart. Then the next thing is how there is the bonding between these two. So the strongest bond is like a glue. The glue has a strongest bond. So just like we will remember crazy glue, it's a 3 hydrogen bond that's between cytosine and guanine. And in adenine and thiamine or uracil, it's always a double bond. Double bond. So it's a very important thing to remember. So firstly nucleoside and nucleotide, then second thing is what are purines and pyrimidines, and third is their bonds, like which bond exists. And one more thing in this one is, so remember, if a nucleotide DNA bases are A and G, and in pyrimidine it is C and T. In RNA it's A and G, but whereas pyrimidine change in RNA to uracil and cytosine. And this thing we have to remember. This question is usually asked in exam. How gout is how gout is caused, like what's the etiology factor of the gout, or what is observed with gout? So it's the increase in uric acid. Then we goes to the biosynthesis. So when we have biosynthesis for nucleotide, so this one is the reaction. We don't need to remember the reaction. And this one, target for antimetabolite methotrexate. This thing is important. So the catabolism of nucleotide results in no ATP production. So in this reaction, there is no ATP production occur. And then next move to nucleic acid. So in nucleic acid, they are basically polar and hydrophilic. So they are lipid loving and say they are polar. So they are polar and hydrophilic means water loving and polar. The backbone consists of phosphodiester bonds, and there will be two chains anti-parallel and complementary. So one will run in the 5 to 3 direction and other one into 3 into 5. So, and this is also very important, so you should make a note on this thing. So in DNA and RNA, there is a reaction which is used to differentiate them, which is a Flugen reaction. So it's basically deoxyribose. It's like test it with this reaction so we can differentiate whether it's a DNA or an RNA. Let's move to the next one. So this thing, then we have the important question, that's the RNA, types of the RNA. So let's see type: ribosomal, transfer, and messenger RNA. Second important thing we have to remember is their location. Remember this one, transfer and messenger, their site of synthesis is nucleus, whereas this one is nucleolus, R nucleolus. So most prevalent is ribosomal RNA, and it's in the nucleolus, and other one is the contain anticodon, this one. Least prevalent is messenger RNA. We can just also remember a little bit their function, like the carries amino acid test, and this one carries genetic code. So next is a nucleosome. So nucleosome is consist of DNA wrapped around a histone octamer. So this one is histone. Histones are, this question is usually asked in ADAT, like histones are composed of, so remember Al, that's like arginine and lysine due to their free amino group. Just remember these two things in the histone, they can be tested in ADAT exam. So this one is a DNA organization, so it's not so important. Just we have to remember like the DNA, the enzymes which are involved in DNA synthesis. Just like what's the function of helicase, it unwinds the DNA molecule, and topoisomerase, it secured the replication fork so that when the DNA strands are separated into leading and lagging strand, just like one is like in the leading strand and one is lagging strand, they're opposite in direction. So these are the like how their functions, like some enzymes are joining, some are helping in the DNA production, some in like in breaking, unwinding the strand. So this thing is very very important, the names and like what the enzymes are used for. Remember helicase unwinds, then topoisomerase, this one DNA polymerase, and the direction of the strand. So always remember one strand, like remember leading one, so leading is 3 to 5, so other will be opposite, that's 5 to 3. And ligase means like a glue, ligase join, which fragment? Oh, Okazaki fragments. Then exonuclease, that like it remove the nucleotide primer. Then is a DNA gyrase. So for the DNA gyrase, the suprachiasmatic structure of the replication fork is passed. So this is very important question. Let's revise again the important things. So make notes of this one. So first we should know what is nucleoside and second nucleotide. So sugar present and RNA base, and then phosphate group, type of purine, pyrimidine, their bonds. Then we will see which one is different in RNA and DNA bases. And second is no ATP production is there when there is catabolism of nucleotide. And next we will go to like this thing, how we can differentiate between DNA and RNA by fusion reaction. Then types of RNA, remember only ribosomal is in the nucleolus and it's most prevalent. Let's go to this one. Just make a like important note of this thing, write it down somewhere, so for the final revision, because it's a very very important. Then the names of all the enzymes in the DNA synthesis, what's their function. So then there is another process, that's transcription and translation, and this one we will learn this one also in bacterial, like in the bacteriology, in the microbiology, like the prokaryotes have this ribosome and eukaryotes have this one. So we will learn this one again. So it just sometimes they can test also, but remember 70 is in prokaryotes and 80 in eukaryotes. Then there is this one, polytranscription, removal of intron. Like the two things that occur in during the RNA slicing is removal of introns and subsequent joining of the exon. So it's also important. Only these things is important in this one, in the RNA synthesis. That's the transcription. In ADAT, they usually test the basics. So like how's the transcription work or what it do, right? So it's the RNA synthesis, whereas in the translation it's the protein synthesis. And one more difference in both these two, so it's this one occur in the nucleus, write it down, and translation occur in the cytoplasm. And then there's the process. It's just for if you want to understand, but don't go so deep in this one. It's in the biology. And then this one I would ask to write it down, very very important, because if the initiation code they will ask, so you have to remember it's the AUG, and the termination, remember UAA, UAG and UGA. So this one and histones, very important. Basically they will give us so many examples, they can give you all the four, and then will ask which codon is responsible for initiation. So remember AUG. And then we have point mutation. This one also very very important for this point. Like the point mutation, I was asked in my main exam in this one, like the sickle cell anemia is under which mutation. So basically it's a point mutation, they can ask in this way also, but the other way is like the sub classification of the point mutation, that's the missense. And third thing we have to remember about sickle cell anemia is that the valine replaces the glutamate. So let's go to then nonsense. So first one is the missense. Let's do the types of the point mutation because others can also be tested. So first missense, then nonsense, then transverse transition. So in transverse, purine is replaced by pyrimidine, whereas transition, purine is replaced by another purine. So remember this point by heart. And this one also, wobble effect, degeneracy reside in the last nucleotide of the triplet. So this one is not so important, just if you want to have a look. So wobble effect, just remember a little bit, but mutation is very important, and the codon one also. Let's go to frame shift mutation. They can be also asked this thing, like the frame shift. So it's a depletion or insertion of one or two base pairs. So frame shift mutation. And repeat, amplification of the sequence of 3 nucleotide. The next thing we will do is the reverse transcriptase. So this one, see, so PCR reaction. So why it is used, like the sequence in this one, so it is used for to amplify a target sequence of DNA, and the sequence is the denaturation of DNA sample into 2 strands, then is the annual primers of each strand, copy each strand by heat stable DNA polymerase, and then the repeat. I would it will be a very helpful for you guys if you will make a notes according to the subject. So if like the you are doing biochemistry, take this chapter and write just important point. I told you this is the most important they can test. Beyond this you will not be tested. So to make this, and so in the final day of the exam or that week you can just revise from your notes, you don't need to open the book again. This one also very important, which technique is used to test which thing. So for this one, for the plotting techniques, remember the word snow drop. So just forget the O. For southern is for DNA. Southern is for DNA, means this one, like this. Northern is for RNA, and for Western we do is protein. This is more than enough to remember. Snowdrop. This acronym will help you to remember this. So this is the whole this chapter for nucleotide and nucleoside. Just make sure you revise from this is from the first aid book part 1. Revise, and all the things I have said that they're important, make an important note and try to remember. So I hope that it will make easy for you guys. Thank you.